Welcome, everyone. We are just going to give it one more minute to allow people time to be admitted to the room. We will start in 1 minute. Okay, I think we can get started. On behalf of our program team, I welcome you all to today's Public Health Lab webinar about the Research Opportunity Announcement OT 25004, which was issued on March 6th. I am Dr. Matt Arnegard, the Office of Strategic Coordination Program Leader for this initiative, and I am also serving as the Other Transactions Program Officer on this research opportunity announcement. This announcement invites applications to support the Newborn Screening by Whole Genome Sequencing Initiative, which is a milestone-driven study of the feasibility of a collaborative, multi-state model for incorporating genomic sequencing as a screening tool into the existing state-based U.S. public health newborn screening program. And this funding opportunity is part of the Common Fund Venture Program. So today, we'll briefly introduce the Common Fund Venture Program, and we'll introduce the ROA, or Research Opportunity Announcement, and highlight some of its key points. And finally, we'll have a question and answer session for the rest of the hour. Please post your questions using this Q&A function highlighted in this red box in this ribbon located at the top of your Teams window.
This references a Q&A function shown on the screen.
The NIH Working Group will address as many questions as possible during the webinar. If we can't get to all of them, we'll follow up individually with attendees whose questions we didn't answer. Questions and answers that relate to a broader audience will be posted on the frequently asked questions page for this initiative, and that web address is given at the bottom of the slide.
The web address is https://commonfund.nih.gov/venture/NBSxWGS/FAQS.
And we'll post those questions anonymously so your names won't be included. And we will also be posting a recording of this webinar, including the Q&A session for those who couldn't make it or want to watch it again. And I also want to announce that we had a technical assistance webinar a week ago for principal investigators interested in applying to this funding opportunity. And the recording of that similar webinar geared towards the primary applicants is already available. And you can find it in two ways. I don't know if you're seeing this, but I want to dismiss these. So this is a screenshot of our webpage. Here's the web address.
The web address is commonfund.nih.gov/venture/NBSxWGS.
commonfund.nih.gov/NBSxWGS. And this is the Common Fund webpage for the initiative. And you can find the webinar from last week in two ways. We have the initiative updates here. And so you have a link to the recording of the technical assistance webinar and the slides, or you can go to the frequently asked questions page within this website, and you can find the links to the recording and the slides there. And those are the same places where we will be posting today's webinar and it will take us a few days to do that. We'll get them up there before Friday or by this Friday. Okay, so let's start with an introduction to the NIH Common Fund and the Venture Program. The NIH Common Fund moves the NIH mission forward faster. It does this by supporting a series of bold scientific programs, each of which is catalyze discovery in an area of research relevant to the NIH mission. These areas of research supported by the Common Fund are cross-cutting rather than focusing on one disease or organ system, and they also advance the missions of multiple NIH institutes and centers. The Common Fund programs are designed so that each program will spur subsequent biomedical advances that otherwise wouldn't be possible without our strategic investment. The main idea behind the Venture Program is to do amazing things with modest funding. This new area of Common Fund support provides a framework for the development of short-term initiatives that embrace scientific risk. Proposed initiatives, the Venture Program initiatives must meet the 5 program criteria listed on the left, which apply to the Common Fund as a whole.
The criteria listed on the screen are transformative, catalytic, goal-driven, synergistic, and novel.
To be a venture initiative, we also expect the initiative to be bold. In other words, make a daring investment that has potential for significant outsized impact. We want venture initiatives to be nimble so we can implement them rapidly in response to specific scientific opportunities. And these initiatives are also focused on clearly defined research topics with a limit of 3 years of funding and up to $5 million in support per year for the whole initiative. And now I'm pleased to introduce Dr. Dominique Pichard, who will give an overview of the ROA and explain how state public health labs can participate in this initiative. Dr. Pichard is the director of the Division of Rare Diseases Research Innovation at the National Center for Advancing Translational Sciences. He serves as coordinator of the Newborn Screening by Whole Genome Sequencing Working Group and is also the primary scientific contact for the ROA. Dr. Pichard, please continue with the presentation.
Thank you very much. We could go to the next slide, please. So the Newborn Screening by Whole Genome Sequencing Collaboratory, which I'll refer to as NBSxWGS for the remainder of this presentation, aims to demonstrate the feasibility of a collaborative model for broader genomic sequencing across multiple states to demonstrate the potential for a national newborn genetic screening program that maintains the public health goal of identifying severe conditions that are treatable early in life and can keep pace with the therapeutic development landscape for rare diseases. On this slide are 4 high-level goals of the program.
The 4 high-level goals are: support centralized analysis and interpretation of WGS results for 5 to 10 state public health labs, enable broad access to WGS as a screening tool in the newborn period, focus on a limited gene panel of serious rare diseases with early treatment options available, examine ethical, legal, and social implications, or ELSI, of population-wide WGS in the newborn period.
This initiative has an award budget that is limited to no more than $4.8 million in total cost per year for a maximum of 3 years. No more than 1 meritorious award will be funded. Next slide, please. I'm sure that you all on this call know the background of the newborn screening program even better than I do, but I did want to draw your attention to the bottom half of this screen. These 4 bullets highlight some of the potential impacts of what adding whole genome sequencing to the existing newborn screening program could achieve.
These 4 bullet highlights are: expand NBS to many more diseases with available treatments, interventions, shorten the diagnostic odyssey for rare diseases that are added to the list, improve outcomes by allowing treatments to start earlier in the disease course, make NBS programs more compatible with emerging technologies, for example, gene therapy, gene editing, by enabling rapid addition of new diseases to NBS.
Expand newborn screening to more diseases with available treatments and interventions early in life. Shorten the diagnostic odyssey for rare diseases that are screened for. Improve outcomes for included conditions by allowing treatments earlier in the disease course. And make the newborn screening programs more compatible with emerging genetic medicines by enabling rapid addition of new conditions to the newborn screening program as treatments become available. This initiative will support a collaborative, multi-state model for newborn screening that would use whole genome sequencing platform to support a first-tier screening assay for a set of genetic conditions that are actionable in the first year of life. Next slide. So to carry out the feasibility study, the NBS by WGS Collaboratory will need to consist of the components shown in this figure.
The infographic shown on the screen is available at https://commonfund.nih.gov/venture/NBSxWGS.
In the funding opportunity, or the ROA, the components are called functions. So you may hear me refer to as functions throughout this presentation. This infographic, as you saw, is posted on the web page, and the address is at the bottom right of this slide.
This infographic can be found at https://commonfund.nih.gov/venture/NBSxWGS.
So I wanted to highlight the 4 core functions of the collaboratory, which are indicated by the rectangles on the bottom and the middle of the slide. And in the upper right corner is the state public health laboratories, who are a critical part of the success of this program. But here I really want to give you a broad overview of the entire program. So I'm going to go through the details of each of the 4 functions of the prime recipient, just so that you have an overall understanding of the roles and relationships between the different components of this collaboratory and how the PHLs fit in. So function 1, the bottom middle with the green outline, is the project administration function, also called the Central Coordination Core. This will be led by the prime recipient. And the function will provide general oversight of the collaboratory, keep track of budget spending and project milestones, manage meetings and communications, receive variant calls from the sequencing and analysis function, use those derived data to assess whether participating newborns are screened positive or negative, and provide general coordination across the study, including coordination among the 5 to 10 participating state public health laboratories. Next, there is the Community Advisory Board on the bottom left with the purple-blue square, and the ELSI function, which you can see encompasses the entire program in the blue-gray square. The Community Advisory Board must be formed within the first 3 months of the award being issued, and should include the perspectives of parents and guardians of newborns, community leaders, healthcare providers, genetic counselors, newborn screening advocates and professionals, bioethicists, and data privacy experts. And the reason that it's important for this board to be formed early is in order to provide guidance on much of the study, including participant recruitment, the informed consent process, the target gene list, the return of the results process, privacy safeguards, and the data management and sharing plan. This function is also responsible for an ELSI research study that will examine the impact of NBS by WGS on newborns and families, state newborn screening programs, or public perceptions of newborn screening. The participant recruitment, consent, and return of result function at the center in the light blue outline will develop participant recruitment plan in communication with the state public health laboratories, develop an informed consent process with input from the Community Advisory Board, communicate with the state PHLs about the transfer of dried blood spot punches from participating newborns to the collaboratory, oversee the return of screen positive results to participating families, and guide participating PHLs on referral and clinical workup recommendations for the screen diseases. Lastly, the sequence and analysis function in the lower right will determine the target gene list with input from the CAB, and will also run the genome sequencing pipeline for all consented samples. They will be responsible to securely store the genomic data during the study, so the data can potentially be reinterrogated if the target gene list is optimized during the study, and they will transfer the derived variant calls to the administrative function or the central coordination core. Next slide, please. The overall purpose is to support a milestone-driven study with significant community involvement that will assess the feasibility of a collaborative model for incorporating whole genome sequencing into the existing state-based U.S. public health newborn screening program. The NIH working group will generate a roster list of public health laboratories that are willing to participate in the collaboratory. During the award negotiations, the potential awardee, together with this NIH working group, will select the 5 to 10 state PHLs from this roster that the NIH working group is developing, as I mentioned. The Community Advisory Board will provide input to guide the development of the informed consent process, list of genes to screen, return of result processes, and the genomic data sharing plan, as I mentioned. Next slide, please. The anticipated work products for this collaboratory from the prime recipient are screening of participating newborns by whole genome sequencing in 5 to 10 states that have different levels of experience with NIH-funded research and/or with whole genome sequencing, to conduct an embedded LC research study, and to generate a final report. If successful, the study will demonstrate the feasibility of adding whole genome sequencing to an existing public health resource available to all babies born in the U.S. The initiative will provide federal and state partners with critical data needed to inform sustained implementation of NBS by whole genome sequencing. Next slide, please. Some of you may not be familiar with other transactions. So just to provide a little bit of an overview, because other transactions are not grants, cooperative agreements, or contracts. So those who may have participated in NIH opportunities that are grants or cooperative agreements, this is a slightly different. Other transactions allow flexibility to adjust the range of activities, milestones, and deliverables based on the objectives and needs of the program. This means that tasks, partners, expertise, and so forth can be added or removed based on changing research landscape or to address unexpected roadblocks. Many NIH policies apply to other transactions, though not the traditional policies that govern grants or contracts. Award funding is different from typical NIH grants. For example, other transactions allows for variable segment lengths, no future funding commitment, and early termination or extension by NIH depending upon the circumstances. Changes in the project and budget are also handled differently. For example, NIH can propose changes beyond what is required in the funding opportunity or ROA, as well as established unique terms and conditions like thresholds for prior approval that is customized for each individual award. Next slide, please. The prime recipients, the applicants are required to submit a draft data management and sharing plan with their application that will outline and justify any potential limitations needed to protect the privacy and confidentiality of newborn participants. But the final data management and sharing plan will be developed in consultation with the Community Advisory Board and the NIH Working Group. Next slide, please. It is our goal for the collaboratory to include 5 to 10 state public health laboratories. In order to achieve the goal of assessing feasibility, it is important to include public health labs that have varying degrees of experience with research, particularly NIH-funded research, as well as varying degrees of experience with whole genome sequencing, from none to proficient. The minimum expectations for state PHLs that participate are to prepare and send consented dried blood spot punches to the central sequencing lab, assist the prime recipient with the process of returning screen positive findings, and assist families and caregivers as well as primary care providers with local referrals for diagnostic confirmatory testing. Next slide, please. The collaboratory will include a community of practice that is developed for the state PHLs that are participating. The community of practice will provide resources and education on topics such as incorporating whole genome sequencing data into PHL workflows. And as you can see on this slide, I'll give you a few seconds to look at the bullets that are listed that are some possible topics that would be covered in the community of practice.
These topics are evaluation of candidate gene lists, variant interpretation, best practices for return of results, processes for adding new genes or variants, troubleshooting WGS workflows or pipelines, reducing implementation barriers.
Next slide, please. As discussed earlier, there are 4 functions that will carry out the work for the collaboratory, each with different expertise and responsibilities. Function 3 for the prime recipient is the whole genome sequencing and analysis function. And I want to highlight from the funding opportunity that one of the responsibilities of this group is to determine the target gene list. The initial list of target genes will be screened will be limited to genes for which genetic variants are known to cause monogenetic diseases with early childhood onset and for which interventions or treatments are available. The final list will be developed with input from the Community Advisory Board, as well as other relevant parties, including the public health laboratories. All participants in the study will be screened for all conditions that are ultimately on the target gene list. Next slide, please. We are inviting all state PHLs to participate and be included on the roster, and we encourage you to email the NIH working group to be included. Being included on the roster does not obligate a state to participate at this moment. It is just demonstrating interest to be on this roster, to be considered for the final 5 to 10 PHLs. We do recognize that each state PHL will have a different set of requirements in order to participate. So we are asking those PHLs that are interested in participating to be familiar with those requirements as well as the timeline anticipated, and that can be communicated to us. It is our hope that the subawards between the prime recipient and the participating public health laboratories will occur in a 6-month time period. But we do not want this to be a deterrent, so we invite you to reach out to the NIH working group to discuss the requirements for your participation if needed. So this is your call to action. The first step for PHLs is to contact the NIH working group at the email listed on this slide.
The NIH working group email is NBSxWGS@od.nih.gov.
to let us know of your interest in being placed on the roster of potential state PHLs to be selected by the prime recipient with input from the NIH working group. Again, this is not a commitment at this point. And please note on this slide that the deadline to be included on the roster is Monday, June 2nd. Next slide. This timeline overview shows the key events and due dates leading up to the earliest possible start date for the awarded project. Again, I would like to highlight the deadline to be included on the roster of public health laboratories to be considered is June 2nd. Another important date is the earliest start date of September 1, 2025. Next slide, please. So now we invite you to ask questions about the funding opportunity and in particular participating as a state public health lab. You can use the Q&A function in Teams, or you can raise your hand and we'll let you know when to come off mute to ask your question. I see that there is a hand raised. Rajesh Sharma, are you able to come off mute? Kristen, could you help in facilitating that?
Can you hear me?
Yes.
So this is Rajesh from California, lab director in the public health laboratory. So my question is, before we put in the roster, we really want to see how many genes will be added to the panel, because that is and how we will basically guide our physician for the follow-up also.
Thank you. So your question says that before you get added to the roster, you would like to know how many genes are going to be included in the final list.
Yes.
And we do not know that because we at NIH are not determining that final list. We really want this to be a list that's generated by experts in the field, including our, which will include our community advisory board, as well as input from the state labs. So that is not something that we are able to provide up in advance.
Okay, but do you have any idea it will be less than 100 gene, or it will be more than 400 genes? Because there are many studies from different other countries like Australia and UK. I think the panel is very limited, so that we are, at least if we have some information that will guide us, because it's not about testing, it's more about how we'll do the follow-up also for
Absolutely.
These disorders.
That is yes, and this is not intended to be broad. Let's get every condition that we possibly can, because again, it is intended to fit within the mission of the newborn screening program as a public health program, and therefore we really are narrowing it down to those that do present early in childhood, as well as those that have a treatment, and so it is going to be, on the smaller side of the ranges that you provided, I anticipate. Okay. This is not intended to test everything. And then we do know from the sequencing programs that have been running through academic institutions and other research endeavors of the percent positivity, depending on what's on your list, we're anticipating about a 2% positivity. So that may help give an understanding of what you may anticipate in terms of workflow for any given state.
Okay, thank you so much.
Yeah, thank you. And I see Anne has her hand raised.
Am I unmuted?
Yes.
Hi. So I put some of my questions in the chat, but I'll ask them. Can you speak to how it is that the public health labs actually have a voice in the development of this collaboratory? Because I don't really understand who the NIH work group is, or how we have a voice in the disorders that might be included and align with our state's criteria for newborn screening, how we have a voice in what our state would like to see at a minimum for consent, or how our state would like to develop our technology. So I think it all also ends with, do you anticipate a transfer of technology to the state public health laboratories, or is this solely for the development of one laboratory in the nation?
Great question. Melissa, could I invite you to address this?
Sure. So, Anne, I think you had about 4 questions in there. So I'm going to try to start with the first one, and then you can tell us what we haven't answered yet. So okay, I heard a question about how does a public health laboratory have input into the strategy, including, developing the list of genes. So again, this is a collaboratory, and this is different from traditional grants and other mechanisms in that as an OTA, we expect that there will be some negotiation that happens and discussions that occur with the prime recipient for this award, as well as the public health laboratories that will be a part of the screening process, as well as the community advisory board. So the key is that the prime recipient is going to be organizing the interactions so that the ultimate list of genes, for example, that are selected for the panel reflect the consensus to the best of the ability of the various participants that agree that these are going to be the key genes and variants that have the highest probability of success, that are the most straightforward to return, and that are actionable within the newborn period.
It's not clear to me how whether the prime recipient, what their responsibility is to the public health laboratory.
Well, they cannot do this project without working with the public health laboratories that will be a part of the group.
I recognize that. Yeah, thank you. I recognize that. It's more, how do the public health laboratories exercise their voice? I mean, do we have a seat at the table, and where is that seat?
Yes, the public health laboratories will be a part of the collaboratory. So this is a group effort that requires coordination, and the coordination function of the prime recipient will be facilitating the communication between the various entities.
Okay.
And I think that the same also applies to the consenting process and the requirements for consent. I mean, each of the prime applicants needs to propose a method for consent, but that will need to be refined and further developed during the negotiation period and with the input from the public health laboratories.
Okay. Do you expect one method of consent across the nation, or possibly multiple methods of consent?
I don't think we have an expectation one way or the other.
Okay.
There could be more than one method.
Can you speak to the eventual transfer of technology to public health laboratories, or what is the end goal here?
Do you want to take that one, or do you want me to?
Sure. Really, the end goal is to look at the feasibility of incorporating whole genome sequencing into the newborn screening program. So this particular research project is not to develop new technology or improve technology. It's really to assess, again, it's to assess feasibility, knowing that implementation, if this were to be, sequencing were to more broadly be incorporated into newborn screening, is not something that happens at the conclusion of this study. This is one portion of answering some of the questions that are needed for future implementation. So I don't have any answers to what happens to technology, because this is not a technology development funding opportunity. That's not the focus of it.
I guess I'm trying to figure out whether the end goal is to ensure that the best and least expensive technology can go forward from public laboratories, or whether there's already a decision to have a single central laboratory in the nation do whole genome sequencing. So there's so much strategy in between those questions, right?
That is correct. There's a lot between kind of what we're looking at here and the ultimate implementation and the strategies around, is there one center for all of the US? Are they regional? Is it individual? And this I would say that this research project is not intended to answer ultimately be the final answer of should there be one lab for all of the US versus not, but rather, looking at a collaborative model, which, may vary depending on ultimately what the implementation looks like down the line outside of this research study, what the final recommendations, from the federal and state partners that are outside of the NIH. We conduct research and we're really asking a lot of the questions to help in the understanding of this feasibility. So as this project, we wanted to look again at kind of doing it collaboratively so that it wasn't that every lab participating had to have the capabilities to conduct the sequencing themselves, because that in and of itself would limit who could participate and doesn't help us answer what are the workflows that are, needed and some of these other feasibility questions. So I hope that gives you a better sense, Anne.
It does. It definitely sounds different than what I heard the other day. It sounds more open-ended.
And the goal here is to create a community of practice where we'll actually learn from the process of doing this in different settings, kind of best practices. It may not be one size fits all, however. We recognize that states have different screening approaches, different parameters for how they do their screening.
Good.
Thank you. I do see we have a couple more raised hands, but we've also gotten several questions posted in the Q&A. So I'm going to pull up some of those questions and then we'll go back to raised hands. What is the anticipated size of the effort, number of enrolled parents, number of those that get sequenced? And the answer to that is the number of enrolled infants who are sequenced is really dependent on the budget available and the overall project plan. So that is something that the prime recipient will ultimately determine based on all the aspects of their proposal. Another question on, can consent be obtained after the blood spot is drawn for newborn screening? And the answer would be yes, this would be possible. However, the consent timeline will be proposed by the prime awardee in their application and should be finalized during negotiation with input from the Community Advisory Board and input as well from the public health laboratories that are participating. Any consenting process proposed really should accomplish the aims of the programs in a timely manner. Are you prepared to work with state PHLs that only have an opt-out process for newborn screening consent or do not have an existing consent interaction with parents? What difficulties would you see in working with these states? And as mentioned in the webinar, we really do want we want public health laboratories with different levels of experience and different consent protocols, to look at what may be potential barriers for implementation or considerations in the future if if we get to implementation outside of this research study. Are PHLs required to be screening programs with access to blood spots, or can other entities such as academic medical centers in collaboration with state screening programs apply? Because the goal of the program is to assess feasibility of adding sequencing into the workflows of the public health labs, to the extent that the PHLs work with other entities, those other entities can be involved. Should we go back to hands raised? Let's see who we have. Rajesh Sharma. I see you have your hand raised.
So, yes. Can you hear me?
Yes.
Yeah. So Rajesh from California again. few questions. First of all, how do you anticipate the role of current biochemical testing in conjunction with the sequencing? And second, will the sequencing will cover only the pathogenic variant, the panel data, NIH in the research, or it will also cover other VUS also? And third, I will say, moving forward, and also has raised a similar point, how NIH anticipate training our current staff and on the technical and moving forward, because this 3 years is a good time period, because if any way NIH will provide funding to train the existing staff, public health staff, basically to gear up for the sequencing assay also. Thank you.
Thank you for those questions. Melissa or Molly, would you like to address?
So I can answer at least one of those. Again, there are several questions embedded. So one of the questions was what types of variants would be reported. So our goal here is to be very definitive about, or as definitive as possible about the variants that are reported as those that are either pathogenic or likely pathogenic. We would not be returning variants of uncertain significance. And we would also anticipate that there would be an orthogonal method for confirming those findings in a CLIA or CLEP certified laboratory, and that those confirmatory tests would then be, there would be a system developed for returning those results in partnership with the PHL laboratories to families and healthcare providers. I think I answered a part of your question, but I'm not sure if I answered the whole thing.
Yes, the training part, I think how NIH thinks that moving forward, how we can train our existing staff, and because that will be a big challenge. And we are, as public health will be doing sequencing, I think we might have a bandwidth, but I think bandwidth is not, and we were already discussing internally, it's not about what technology can provide, but we have to partnership with our clinician also, because right now what I'm hearing, at least in California, they have a very hard time in even taking care of the current workload also. And so what they really want from us, when we send any positive to them, they want that should be real positive. At least they are not worrying about the false positive. So where I think that sequencing can help us is really filling that gap, basically. So when we are sending to the clinician, we are sending true positives. So that will be very helpful. But again, we don't know the disorders. What disorder will be added in that list, if it is diabetes type 1 or something that is really, follow-up is really much more challenging, what I'm hearing. So first the gene list, and then working with our partner in the clinician, because they are the key. I think we can do anything we want in the public health lab. Sequencing technology is very good. We can even add 400 gene or more than that also. But again, it is a clinician who will be basically the, so we really want to have that conversation with the clinician also. So again, the same question, along with the public health laboratory, are you involving some of the biochemical geneticists clinicians or other clinician who will be, because it is a full partnership, I think that has to play here.
Absolutely, and I think this really raises some of the complexity around, some of the screening, and I think to address, the question about maybe some of the training, that's the community of practice that we've built in, to provide opportunities to train, to provide that education, be a resource to the labs. So we hope that community of practice will be a model to really provide some of this background. But also, as you are stating, with referrals to, within the state, and these will be new conditions that are added. And so as part of the participant recruitment, consenting, return of result function, which is function 2, there is a clinical referral centralized resource, which will guide the state PHLs on appropriate referrals and clinical workup recommendations for those conditions that are included on the gene list for when they have screen positive participants. So I hope that helps to address those questions. And oh, go ahead.
No, thank you so much.
Yeah, let's say related to that, we got a question in the Q&A about will or should the initial gene list include RUSP conditions? And we, as I mentioned earlier, we don't have a priority, all of the, we don't have that list of conditions because we want this to be something that is developed with input from those with deep expertise, as well as lived experience. But ultimately, we anticipate that this will be new genes or conditions outside of what is currently recommended on the RUSP, to really look at, because if there's already a good model, then this is to look at additional genes or conditions. But really an argument could be made to add genes where sequencing confirmation could significantly enhance the diagnostic yield of metabolic or enzyme screening that currently exists on the RUSP, but it's not intended to duplicate the conditions on the RUSP.
Should we go back to the hands raised? We have,
yes.
Next up is Tulsiram Prathapam. Sorry if I'm mispronouncing your name.
Yeah, good. Yeah, my name is Ram. You can call me Ram, and I'm from Illinois Department of Public Health. My question is, in order to become a participating laboratory, is this mandatory to send the specimens? Because I'm not sure whether we have informed consent for using the specimens for research. That's my main question. What are the other ways PHL can contribute to the become a participating laboratory? For example, I'm a board certified molecular geneticist, so I could help you in the vision of helping preparing the gene list and the variants, potential significant variants. I can be I can participate as a part of collaborative from the Illinois Department of Public Health. Is this mandatory to send the specimens or not?
I can answer that one. I mean, so this is the requirement for this effort is as a research project is that because it is research, there will need to be informed consent obtained for those families who are participating in the study. Now, what that informed consent process will look like may vary among the states, and we don't know exactly what that is going to look like at this point, but we're asking the prime applicant to propose an informed consent process that the states would need to incorporate in order to participate as one of the 5 to 10 public health laboratories that are part of the collaboratory. There may be other opportunities outside of that participation for folks such as yourself, biochemical geneticists, bioethicists, many others, laboratorians who have expertise in newborn screening that may provide expert opinion and participate in the community advisory board activities, but we don't know yet at this point what that exactly will look like.
Okay, thank you.
Look at submitted questions. Who will lead the submittal to the FDA for approval, or will each lab be creating a new 510K IVD? And this will be the prime recipient's responsibility. So this will not fall to the state lab. And will we know who the prime recipient of the award will be before determining whether or not we want to participate in the program? We are collecting, we are developing the roster. And so at the time of putting any given public health laboratory on the roster. The application cycle is still going, so the prime recipient will not be known at that time. And then during the negotiation period where the final 5 to 10 state labs are selected, depending on the time of the award and processing of the onboarding of states, it would be anticipated that before finalizing the commitment as a public health lab, you would know the prime. And then a question about will IRB be needed to participate? If so, will you all start that process? And because this is a research study involving human research participants and data, an IRB approval will be needed. And that is not something that the NIH will start, but the prime recipient. That is in their roles and responsibilities to get a protocol that is IRB approved. I'm looking through these.
Why don't we go back to the live questions while you're looking through those? And
okay.
Next up, we have Kimberly Noble Piper. I think you're next. Are you still? Can you unmute and ask your question?
Yeah, I think, can you hear me now?
Yes.
Sounds like, okay, great. Thank you. I have 2 questions. Hopefully they're quick. So newborn screening is a system, correct? It's not just public health laboratory. I was just wondering if the expectation is that if the application to be included on the roster needs to come from the public health lab component of the system of screening, or if it can be from the entity that has the authority to administer the program.
Yes, I would say it is the latter. So
okay.
It would be from the entity that has the authority to administer the program for any given state.
Okay. And the second component is who bears the costs. So will there be costs to the participating families? Who bears the costs for the public health laboratory or the systems that are doing the, setting up the system to be able to follow up on the return results and those kind of things? So.
Yeah, so in terms of the cost to the entity that administers the newborn screening program, that would be budgeted in the application. And ultimately, the prime recipient would have some budget to cover the expenses of the newborn screening program or state lab, however that's administered. In terms, I believe the second part of the question was in terms of cost to participants. And Melissa, I'll hand that over to you.
We would not expect any cost to the participants because the research study itself should be covering the costs. There may be costs associated with confirming a diagnosis through clinical or biochemical means, which again, we anticipate should be borne by the prime recipient through subawards to the state screening programs or entities that administer the state screening programs.
Great, thank you.
I think a little bit related to that, so I'll jump in with this one. What is the expectation of returning results to parents, and will materials be developed on each of the disorders? So for the first question, screen positive results are going to be returned to the parents or the providers in order to have the actionable results to that family. And it is the prime recipient that would develop the materials for sharing with the state PHLs for the return of results, and it will be comprehensive to include each of the disorders that are included on the target gene list. There's a question on to participate. Is there a specific number of specimens or birth facilities that would be required to submit? If we are able to get 1 or 2 hospitals to work with us for obtaining consent, would that be sufficient for submitting specimens for testing for this project? There is no minimum number of birth facilities, but really the goal of the program is to screen statewide in order to have a representative sample from the entire state, as well as understanding that workflow in terms of, again, what are the barriers in this process. There's a question. Do you anticipate that PHLs will continue their regular screening methods for some of the disorders? As I imagine there will be overlap. If so, will you collect data on time to reporting to PCP diagnosis compared to those more standard methods? And I addressed part of this earlier for another question, but this project will not, should not interfere with the traditional newborn screening that each state currently conducts. We absolutely do not want to have a negative impact on the newborn screening programs as they currently exist while we're doing this feasibility study. So we expect that will continue as normal with all the participating PHLs. The prime recipient could propose to collect data on time to reporting, but that is not a requirement of this funding opportunity. For different reasons, we didn't make it a requirement, but certainly it could be proposed. And the prime could propose to look at traditional biomarker screening relative to whole genome sequencing-based screening. Again, just not a requirement.
Shall we return to the raised hands? I just want to remind everyone that we didn't get to a few of these questions, and we will be individually following up and also posting answers on the FAQs. But I wonder if we could maybe get through. We have 4 raised hands. It's possible we could get through them. So, Anne Como, do you want to come off mute? Sorry, Dominique.
Oh, I was going to say, should we allow Scott to ask a question since he hasn't asked a question yet? But yeah. Anne, you already started talking, so please go ahead.
Thank you. Hopefully it will be quick. It's interesting that you say statewide, because I was wondering about that, and I actually have a posted question about costs. I'm hoping that you are anticipating the significant costs to the laboratory information management systems that the public health laboratories will have to embrace in order to make sure that the retrieved specimen is the one that has the consent, and in order to make sure that the appropriate results are going out, and for us to pull together our clinical working groups that we typically work with. I honestly was hoping that you would say something about the number of specimens. I mean, it's a very big difference between 50 to 100 specimens versus statewide.
So I can answer part of that question. We would anticipate that some of the costs that would be incurred by the state laboratories would include potential upgrade to LIMS systems and ability to functionally process the samples and connecting, as you say, consented samples with outcomes and results. We also, when we say statewide, that doesn't mean 100% of all of a state might necessarily be participating in this, because we recognize that under the constraints of needing informed consent, that may provide some limitations on an ability of a state to completely ascertain all of its infants through this mechanism. However, we think that as many facilities as our birthing facilities as feasible would be ideal, because again, we're trying to get as representative a sample as possible from a given state, recognizing that 100% of the babies in each state are not going to be able to be fully consented for this initiative.
Okay, thank you. That's an answer. Good, thank you.
Let's go to Scott. Scott, we're going out of order here.
Okay, thanks. So I just wanted to comment that I appreciate that the request is for public health labs to sign up for the roster. But I did want to point out and sort of channel Kim Piper a bit that newborn screening is a system, and a large portion, perhaps the most complex part of what this feasibility study is the follow-up. And so I think it is important for public health labs to realize that they should not roster unless their follow-up programs. And for example, in North Carolina, our follow-up program is in a completely different division. And so for me to say I'm interested merely gives the prime access to spots for sequencing, but there's a whole large portion of this study that is going to be contingent on follow-up. And you might want to assure that at some level of that rostering process is that labs are speaking on behalf of the program and not just the lab themselves. Thank you.
That's a great point, Scott. Thank you.
Yeah,
totally agree.
And then continuing out of order, and then we'll come back to Rajesh. Let's go to Kendra. Kendra Croak.
Hi, can you hear me?
Yes.
Okay, perfect. My question is relatively quick, and it might have been stated already. So are you anticipating then that this might be an entirely separate sample that would be drawn, or that we'd be splitting the samples that we already collect, and you would get like part of our cards? Like, how much sample are you anticipating needing for this project and with the consent process, and not trying to interfere with our current processes? I'm just trying to figure out if this is going to end up being something where we have to end up drawing an entirely separate sample for this in addition to a normal newborn screen, or if this is something where we can send you like, you know, a single spot, provided that we have one available.
So I just to answer that question, so I think that's a really excellent question. The expectation is that this would be incorporated into the current newborn screening workflows. So we would not expect additional blood samples to be obtained from the newborns, but expect instead that punches from a dried blood spot would be sent to the centralized sequencing facility for the analysis.
Okay. And it is just one centralized sequencing facility that you are considering that would be the prime recipient. Is that correct?
That is correct, for the purposes of this feasibility study.
Okay, that's all I had. Thank you very much.
Raj, you get the last question, and it needs to be quick. And then we will do follow-ups on our fax page to everyone who asked questions anonymously. So go ahead with your final question.
Yeah, Matthew, I think my question is also related to consent. I think it takes 15 days to a month to complete the study at our end. And so we were thinking how, while we are collecting the sample on the prospective, can retrospective sample can also be consented also, because while we are doing this, it takes 15 days, 1 month, maybe can we go 1 or 2 months back samples, and we can easily get the retrospective, consent on those one, and it can easily send you those cards also.
Okay, well, thanks for that perspective. I was seeing if Dominique or Melissa wanted to comment on that retroactive consent.
I would say that, you know, the prime recipient can propose what they consider to be appropriate, you know, of course, in the you know, with the perspective of the goals of this program and feasibility. But if a retrospective sample from the prior, I think you had said month or so, you know, or some period of time, if that was something that the prime recipient wants to propose, it can certainly be considered. Melissa, did you have additional thoughts?
No, I agree with that. I don't think we want to be ascertaining samples from last year's collections or babies born a long time ago. It really needs to be at least temporally related to the ascertainment of the initial dried blood spots. But I do think that what the prime recipient proposes, as long as it's compatible with the consenting process, should be considered.
And follow-up question on that, I think, do you are looking for a diverse group of samples, or do you have any idea in mind? Okay, I'm looking for to have a big, because like California population is very diverse, and do you are looking for a specific group of sample? Okay, I want from this ethnicity, I need this group many sample, and this ethnicity. I know it is very complex question. I don't, but anywhere I think that. You are collecting the diverse group of samples.
We would like to see a representative collection of samples from the population in your state.
Okay. Thank you so much.
Well, I'll just close by thanking everyone for their participation and their interest. And it's exciting to see so many people, so many labs here represented. And in the interest of time, we are going to have to cut it short now, but we will post answers to the questions that we didn't get to on the FAQs page. So thanks again for attending, and have a great day, everyone.